So let us continue in our journey. So what happens, now that we've talked at length on how to make the antibodies, this whole exhaustive process, what happens when the antibody runs into the antigen AG, antibody AB? What happens next? So we have this process, right, where the antibody could be binding two different sites on the same bacterium or whatever it is, but it could be binding two different molecules. And so that process is agglutination. And what is the antibody that's best suited for this, most likely to cause this, of the different types that we talked about? IgM, right? Why? Because how many different Y's are there in that structure? There are 5. It's a pentamer. Their valence is what? 10. Okay, so they can bind 10 different things. So they're going to make these big complexes. So there's agglutination. That can happen. There's activation of complement. So remember that antibody binding to its antigen can bind to some of the complement proteins. We didn't talk about exactly which ones, and we don't care. But ultimately, that leads to the activation of what? C3, which chops it in half. And that leads to 3 outcomes: inflammation, opsonization, coating, right? And making holes in the membrane. So we have opsonization, we have antibodies coating this guy, right? Consequently, what's happening in this picture once he's coated? It really triggers those phagocytes to gobble up anybody that's flagged like this. We also have neutralization. Okay, so what's happening in this picture? These antibodies are binding to this epitopes on viruses or epitopes on a bacterial capsule. And what does neutralization mean? We're neutralizing the ability of this virus or this bacterium or this toxin to do what? To bind whatever it's going to bind in your body. So normally these pieces would be free to latch onto some cell. You know, the virus would trick the cell into taking it up, but now it's covered in antibodies, it really can't do that. Or maybe a bacterium has some kind of attachment, or maybe a toxin, right, would normally have some effect, but now it's covered in antibodies. So it can't bind to whatever it was going to bind to and do the work it was going to do. And then there's this antibody-dependent cell-mediated cytotoxicity that we'll talk about that involves a variety of different cells kind of working together to kill something big like a multi-celled parasite. So agglutination is helpful because it reduces the number of infectious units. So initially when these were floating around there were 5 of them, but we have these antibodies binding them in a clump. So now they're all together, and that means it's going to be easier to do what? Phagocytose them, right? Because they're all together, they've been sequestered, and then we can have just a couple of phagocytes can come along and deal with this as opposed to having to have 5 different guys. Opsonization, why is opsonization helpful? Covering the surface of the microbe with an antibody is doing what? It activates those macrophages, it activates those phagocytes, it says this is the guy we know for sure you want to be dealing with. Helpful to, if you're an antibody bound to your target, why would you want to turn on the complement system? Because it turns on those 3 things. It's just like a switch, and now 3 different effects go all out at once. We flag guys more easily, we activate macrophages and other phagocytes, we cause inflammation, we put holes in membranes. This we'll talk about in a second. In a little while. Neutralization we've already talked about. So B cells, which we just spent a lot of time talking about, the major point of having B cells is to do what? Make antibodies. We have T cells, the whole other half of this. We already know some of what T cells do. So already we know what about T cells? T helpers, specifically what do? They talk to the B cell. The B cell's, hey, should I be making antibodies? The T helper says, why, yes, you should. Go right ahead. Okay? So that's not all, okay? But let's compare T cells and B cells, generally speaking. Where do B cells mature? bone marrow. Where do T cells mature? In the thymus. Now, T cells come in two flavors, T cytotoxic or T helper. Both of these, if the T is first, TC or TH, they're considered naive or immature. They have not yet been activated. In this state, these two kinds, in the thymus, they are educated. They're screened, basically. So any T cell that reacts against endogenous antigens, like normal things in your body that we don't want to be attacking, those T cells are killed. So there's this thymic selection that eliminates many of the T cells because we're creating T cells that are they have a whole variety of specificities. And at the outset, there's no selection against things that would react against your own body. That happens in the thymus. It's a really important part of making sure that you don't attack your own body. Do T cells produce antibodies the way that B cells do? No, they do not. Instead, they do bind antigens, but they use a T cell receptor, or TCR. Now, like antibodies, TCRs are on the surface of the T cell, and on that T cell, just like on the B cell, that T cell receptor, which maybe looks like, instead of looking like a Y, maybe it looks like just a block with some kind of specificity on the end. But all of the receptors on this particular T cell are looking for circles. And on a different T cell, all of the cell surface receptors are looking for squares, right? So the specificity on each cell is different and it's unique. So all of the receptors are going to look for the same thing on that T cell. It's got a very dedicated job. Now, unlike antibodies, a really important component of the T cell receptor, it's not bivalent. It only has one binding site. Now, what do we mean by free antigen? Free antigen is an antigen that's floating around in your bloodstream. So it's the surface of some bacteria, it's a toxin, it's a viral particle. Did T cells interact with the stuff that's coming into your body, and is it sampling those bacterial capsules? Is it talking to those viral particles? Is it binding directly to toxins? It's not. Where is it finding its antigen? It's looking for it on the surface of antigen-presenting cells. So we'll talk about that. Antigen is presented to T cells by antigen-presenting cells. So where do they find their antigens on the surface of these APCs? They look for them on little baskets that are on the surface of special parts of your immune cells called MHC1, Major Histocompatibility Complex 1, or MHC2, Major Histocompatibility Complex 2. And which one of those basket types is found on the surface of a B cell? Type 2, okay? Now, MHC2s are bound specifically by T helper cells, okay? T helper cells are looking in the MHC2 basket, which is why T helper cells are the ones to talk to B cells. And they activate other immune cells. So all of the immune cells in the body that need to be activated, B cells are one kind, there are a few others actually, macrophages among them. They're activated by T helper cells that look in the MHC2 basket. MHC1 is a different basket. That is where T cytotoxic cells look. And that is part of the process of looking for weird stuff going on inside of your body cells. And we'll talk about what that is, but just consider for a second, why might you want to target your own body cells? We've talked a lot about targeting microbes. Stuff coming in from the outside world is not part of your body at all. But what kind of body cells might we actually want to kill? Cancer cells and? You might want to get rid of dead cells. It's not, yeah, that's part of what happens. We clean up dead cells. There's a slightly different program for that. What else? Say again? Bad proteins? But those are going to be things that, generally speaking, it's true, actually. And in a way, you're actually on the right track because Really severe changes in a normal body cell are going to change what's in that MHC1 basket. Most of your body cells are presenting the stuff inside of them on the MHC1 basket all the time, saying, hey, am I okay, am I okay, am I okay, am I okay? Then at some point you might not be okay, and then you're going to get targeted, but most of the time you'll be okay. So it's this constant big brother double checking of your whole body. So we'll get back to that, but you are actually on the right track, and I want you to think about what circumstances besides cancer, which is actually part of the answer. Cancer cells have a lot of mutations, so they make really funky proteins that shouldn't be there. What other kinds of cells might be making proteins that normally you would never make? Under what conditions, what kind of infection, for instance? HIV is a good answer. What kind of pathogen is HIV? A virus. Do viruses cause your body cells to make lots of proteins that are unusual? Like protein capsid proteins, right? Or even RNA-dependent RNA polymerase. Various kinds of proteins are made when you're infected with viruses that would end up on that basket, and the T cytotoxic cells would read that and go, no, we got to get rid of you. So, the ultimate target of the T cell, 1, pathogens entering the gastrointestinal or respiratory tract. And so these guys, most of the time, are getting in our bodies through mucous membranes, and in the process, they're passing through M cells and that are part of Peyer's patches, which contain antigen-presenting cells. So we'll just look at this. This is a part of your intestinal lining, and these are like lots of normal intestinal cells. And right here is what we call a Peyer's patch, and you can see in this cartoon what that looks like. Here are these normal cells of the microvilli that are important for taking up nutrients, and then this sort of wacky cell with lots of little holes, which is a little bit more porous, because this is where your immune system is sampling what's coming into your body, and kind of it gives a concentrated way for all of these immune cells to recognize antigens that might need to be something that we have to fight against. Okay, so going back to T cells, I know this is a complicated diagram and there's lots of stuff on it, but it kind of helps us to understand how the T cells work. So T cells have this T cell receptor. The T cell receptor is doing what? What is the T cell receptor for? It's binding antigens, right? It has some specificity. It's looking for something like this circle. Now, it also has what we call a CD protein, which is another kind of receptor. All T cells have CDs on them. Cytotoxic T cells have something called CD8, and helper T cells have something called CD4. Have you ever heard of these terms before? Probably you have. And our understanding of them in the layperson's world is largely because of HIV. So CD4 is the receptor for HIV particles, and what cell type is CD4 found on? It's on T cells. It's on T helper cells. So that is how HIV sneaks into your immune system. And it's one reason that it's so devastating to your immune system. Now, what is CD4 normally doing? Normally, CD4 is on the T helper cell, and what is a T helper cell looking for? What basket is it looking in? MHC2 on what kind of cells? B cells and other antigen-presenting cells of your immune system, okay? So how does it know to look in that basket, right? It doesn't have eyes. It doesn't have a brain. Our cells talk to each other because they have proteins that are looking for specific things. So this T cell receptor is looking for an epitope. What is this CD4 doing? It's looking for the basket. It helps the T cell to find the basket, and then the T cell receptor can look in the basket. So these work together. So the CD4 is specific to MHC class 2. And so what you can see here, for instance, an antigen-presenting cell, this is a B cell. This B cell has encountered the thing it was looking for, chopped it up, and presented it on the MHC2 basket. And this T helper cell is going to use CD4 to locate these baskets, and then it's going to use the T cell receptor to see if what's in the basket is something of interest. Kurt?
So it'll go off to leave, bind, and then leave, and maybe go to a different part, find some basket somewhere and look.
Yeah, that's absolutely possible. If it finds what it's looking for, then it's going to be a stronger interaction and a longer one, and they're going to start talking to each other with the cytokines and activating, and that's sort of like the end of the story. But if there's sort of, this guy here is going to bind to the basket, and this does not bind to what's in the basket, it's going to get bored and leave, and then might go and visit another basket. All right, so that's what's happening with T helper cells. It's looking on antigen-presenting cells of the immune system, like B cells, and it's looking for what's in the basket. And what's in the basket is going to be stuff that B cell or other antigen-presenting cell found in the body that wasn't supposed to be there, chopped it up, and showed it in the basket. Okay? Now it's waiting for a T helper cell to find it and look in it. Cytotoxic T cells have a similar set of receptors. They also have a T cell receptor that has some specificity. They also have a CD protein, but it's different. It's called CD8. Now, we've talked about MHC class 2. So what do you think CD8 is specific for? MHC class 1. Where are those MHC1 baskets? They are not on antigen-presenting cells. They're not on B cells or macrophages. What are they on? All the cells in the body. Because this cytotoxic T cell is what's scanning your whole body all the time just to make sure that the stuff that is being made by that cell is normal. Okay, so which of these cells is going to protect us against cancer? The cytotoxic T cell. Which one of these cells is going to notice if our epithelium in our nose has been infected with a cold virus and is making all sorts of crazy viral proteins? The cytotoxic T cell. Which one of these is going to help activate an antibody plasma cell to respond to, say, some bacterial infection? T helper cell. All right, so let's look at these T cells in more detail, if you even can believe that there's more to know. The T helper cell is a CD4 plus cell. That means that it has CD4 on the surface besides its T cell receptor. It's important for activating antigen-presenting cells. So it's recognizing antigens in that MHC2 basket. And then the activated T helper cell, so basically on the antigen-presenting cell, and this is complicated, there's another thing that if there's a binding event, then not only is the T cell talking to the B cell, but the B cell talks to the T cell. So there's actually a conversation. It's a two-way dialogue. So not only is the B cell getting a second opinion, but the B cell's telling the T cell also, we're both right. That activates the T cell. So both the B cell and the T cell get activated in the same event. So an activated T helper cell makes cytokines, and in addition to talking to the B cell, it itself proliferates. It undergoes a lot of cell division, and it differentiates into two T helper cells, T helper 1 and T helper 2, and some memory cells. So these memory cells are going to be helpful when? The next time we encounter the same antigen. T helper 1s, they express interferon gamma, which is an important message that activates macrophages and gets them more active to engulf bacteria, because now we know we're dealing with something. We know that there are several other kinds of interferons that we talked about in the innate immune response, interferon alpha and interferon beta. Now, what cells make those two kinds of interferons? They're body cells under a specific kind of pathogenic attack. And it's not bacteria. Virus, yes, if you're infected with a virus, you will make interferon. Now remember what the interferon alpha and beta do in that context. The first cell that gets infected with a virus makes interferon alpha and beta, and then what happens to that interferon alpha and beta? It diffuses locally to the neighbors, and so before those neighbors are ever exposed to viral particles, they're starting to prepare. by making antiviral proteins. Remember that? Yeah. Now that's what Th1 does, and the interferon alpha and beta I just mentioned to distinguish it from what interferon gamma does. T helper cell type 2, these are the guys that activate B cells to produce IgE and activate eosinophils, which help us to combat parasites and also are part of our allergic response if we're having an allergic response. All right, so T cells are often activated by dendritic cells. So dendritic cells are these antigen-presenting cells that run around in the body. They're part of our innate immune response. They're found in skin, mucous membranes, and the GI tract, and they are encountering antigens all the time. Taking them in, chopping them up, putting them in their MHC what class? MHC2 basket, and asking T helper cells what's going on. Now, I just want to point out something that is maybe seems like a subtlety, but is actually important. What do you notice about this picture on the right? There's our dendritic cell, and how many different kinds of MHC baskets do you see? Now, there's MHC2. That one is going to be read by what kind of T cell? MHC2, T helper cells, for the purpose of checking for, is the foreign stuff important? Like, are we going to activate T cells? These guys also have MHC1. Why do they have MHC1? And who is going to talk to that basket? The cytotoxic T cells. Why? Do you just want to screen your kidney and lungs and skin, or do you also want to know if your immune cells are okay? So immune cells have both kinds. Okay? You can kill immune cells. If the immune cell gets infected with a virus, you can kill them too. So dendritic cells are among the antigen-presenting cells that activate T cells. And this is an important way that we also can activate T cells, get more of them, and have ones around that might be able to talk to B cells. So here is an example of activating a T helper cell. And again, a T helper cell has what kind of CD on it? CD4. CD4 is binding to what MHC? MHC class 2. So basically, if a T helper cell encounters what it's looking for, when it's looking in that MHC class 2 basket, then whatever cell it's talking to, whether it's a B cell or a dendritic cell or some other kind of macrophage, they'll have a conversation where the T cell, using cytokines, activates that antigen-presenting cell, and the antigen-presenting cell activates the T helper cell. And T helper cells are really important in this, in activating various kinds of cells, from macrophages to B cells. Okay, so that's what T helper cells do. Let's talk about the cytotoxic T cells. And again, we already know what about these cell types. What is their primary function? Screen the body, looking for, making sure that the cell itself is making normal things, nothing weird. So under what circumstances might we be making something weird? If we had cancer, so we might have mutations where we generate proteins that are not normally made that might create antigens, epitopes, on those proteins that could be recognized as non-self, or viral infection. And in fact, it's also true that if you're under attack by some bacteria that get inside of cells, then as you're presenting things in your basket, you might have bacterial stuff to present to. But usually it's viral. So T cytotoxic cells have CD8. Their target cells are body cells, and they're constantly looking inside the body cell for what we call endogenous antigens. That means our own antigens, the stuff that we're making, we put in the basket. And as long as we're making normal stuff, then there won't be any T cells that recognize that stuff because they will have been screened out in the thymus, in that early education. But if there's anything in the basket that looks weird, for instance, if we have some kind of intracellular pathogen like a virus that helps us to make weird proteins, or if we have cancer, then there'll be something in the basket. Now, a naive T cytotoxic cell can be activated by a dendritic cell and or by the body cell, and when it's activated, it turns into a cytotoxic T lymphocyte. So I know that these terms are very similar. So cytotoxic T cell or TC cell sounds a lot like cytotoxic T lymphocyte. They sound almost exactly the same, right? They are actually different, and I'm sorry. I didn't make this up, but if you see the term CTL or cytotoxic T lymphocyte as opposed to cytotoxic T cell, then that means it is matured and it is actually now going to become this cytotoxic cell. What is it doing? When it binds and recognizes the right antigen in the MHC1, it makes holes in that cell using perforin, a protein called perforin. So this is a lot like one of the branches of the innate immune response, right? One of the branches of the innate immune response uses complement proteins to poke holes in foreign cells. We have a way to poke holes in our own cells. We use a different protein, but it's essentially the same idea. So we make holes, and then through those holes, we shove a lot of enzyme called granzyme that initiates a program called apoptosis, or programmed cell death. All right. So basically, cytotoxic T cells can induce body cells that it doesn't like, such as virally infected or cancerous cells, to commit suicide. And as I said, healthy cells would not be recognized by cytotoxic T cells because those cells would have been screened out, so they wouldn't even exist. But if you're making something weird, then you will be recognized. That will cause that naive cytotoxic T cell to mature into a CTL, or a cytotoxic T lymphocyte, that then makes perforin, poking holes, and then throws a bunch of granzyme in after it, and that initiates a programmed cell death. So here's just a view of what that looks like. So again, we have our body cell. Here's our MHC1 basket. And whatever we put in that basket is going to be read by the cytotoxic T cell, and if it's activated, it turns into a CTL. And now what happens is it's making holes in this guy and causing him to kill himself. This is what apoptosis looks like. On the bottom we have or on the top we have a cell undergoing apoptosis, which basically is this very controlled process of taking apart the whole cell. And these little blebs are membranes. Just basically you're taking it apart piece by piece so that you're not just exploding it, which would cause damage to the neighboring cells, because remember, there are lots of things inside of the cell, and particularly the lysosome has a lot of things in it that are dangerous. You don't want the cell to just spill its contents. We actually are taking it apart in very discrete chunks, and usually then those cells are also engulfed by macrophages or other phagocytes, just the same way you would get rid of a foreign invader. All right. So there's one more kind of T cell that we haven't covered, and I'm just going to mention it very briefly. They're called T regulatory cells, and they have both CD4 and CD25 on their surface. And the only thing that you need to remember about them is that they're an important part of how we prevent an autoimmune disease. So they kind of make sure, remember the T cells mature in the thymus. And one of the reasons that that's important is it's in the thymus that anybody who recognizes the body is killed. So we have tons of T cells basically at random recognizing lots of things, and in the thymus we go and just get rid of anybody that is going to be dangerous to us. But this isn't foolproof, and sometimes a T cell might escape that would actually recognize body cells, and you could only imagine, right, if there's a lot of TC, cytotoxic T cells running around, recognizing very normal things on your body, and initiating that differentiation process, turning into a cytotoxic T lymphocyte and poking holes in all of these cells and killing them all, that would be really bad. So in order to prevent that, we have this safeguard. The T regulatory cells basically look for those T cells that escaped the thymus without being properly educated, and make sure to get rid of those guys. They're also useful in protecting our normal microbial flora, our normal microbiota. And it's thought that they're important in protecting the fetus from being rejected during pregnancy, because remember, we have this creature, in pregnancy you have a creature growing inside of you that is technically not you. So there has to be some way to protect that from an immune response. How do antigen-presenting cells work? We've talked about this already. They take in what they see, they chop it up, they put it in their MHC basket. Which are these cells? Well, B cells, right? We have B cells that put antigens in their MHC class 2s. And which branch of the innate or of the adaptive immune system, of these two branches that we've talked about, humoral or cellular, do we put B cells in? Are they part of the humoral response or the cellular response? This is a total memorization thing that I'm subjecting you to. What do you think? Who votes humoral? Who votes cellular? It's humerals win, not just by majority, but by the fact that that's true. B cell, even though it's a cell, is considered part of the humoral response because its main job is to secrete soluble antibodies, and those are considered part of the humor, okay, the fluid part of our body. We also have dendritic cells and activated macrophages, part of the cellular immune response, that are also APCs. So any of these guys might run into something. B cells are going to run into antigen because they bind it with their antibodies. Then they're going to take it in, chop it up into pieces, and put it in the MHC2 basket. These guys are going to run into all kinds of things and sample them. Right? They just run into stuff, take them in, chop them up, put them in the basket. Maybe they're, important, who knows? Here's what a dendritic cell looks like. The reason they're called dendritic cells is because they have these long, skinny processes, and when they were first discovered, they really reminded people of the brain cells, but they're not found in brain. So they're found in your skin, your lymph nodes, spleen, thymus, blood, and your genital tract, but they're not found in your brain. And they basically, anything that comes into your body, they're sort of a first line of defense in the immune response. They'll just engulf and chop up that thing. Then they will travel to the lymph node where there's lots of T cells wandering around and say, hey, anything in here that you recognize? Should we do something about this? Activated macrophages are another antigen-presenting cell. This is what they look like. Here they are in their inactive state where they look kind of like a pom-pom, kind of, not very scary. This is when they're activated. Okay, they actually turn into what look like little cabbages. So they're physically different once they're activated. They're much more active. They're much more able to find and engulf things. So they have an important job in our bodies to actually take the garbage out. What you were saying before is how do you get rid of cells that are sort of dying? These guys will do that. Anything that's sort of broken down, they will engulf. But they also, if they get a signal to become activated, then they become much more efficient at phagocytosing anything that doesn't look right. Finally, we have natural killer cells. They're very similar to the cytotoxic T cells in that they make holes in cells and they cause them to undergo cell death. They do this with cells that are coated with antibody. And so, for instance, if you're an infected cell and you have a viral protein on your surface, that might cause antibodies to bind to that cell, actually, and essentially opsonize that body cell. The other thing they do, and this is really important, they destroy cells that don't express MHC1 baskets. So just think about that for a second. Who is displaying MHC1 cells? MHC1 baskets. Who is MHC1 on their surface? All your body cells, including immune cells. Now, if you're a pathogen, and remember that the MHC1 basket is there because it's a way for your T cytotoxic cells to read all the body cells and make sure they're okay. So the only way they'd know something was awry is if there's an MHC1 basket. So if you were a pathogen and you wanted to avoid being detected by the immune system, what would be one thing that you maybe could do? Prevent the basket from going to the surface, right? So what do we do about that? We have natural killer cells that go in after the TC cells have screened all the baskets and look for anybody that's not wearing a basket. Okay, and then they kill those guys. So they get rid of anyone who might be missed by the cytotoxic T cells, because all body cells should have an MHC1 for TC inspection. So basically, if you try to go off the grid, these are the guys that are going to nab you. And indeed, some viruses do try this trick. They try to remove the MHC1 in order to prevent recognition by the cytotoxic T cells. Cancer cells might actually forget how to put that basket up, okay? They just get senile, they are busy with doing other things, their DNA is too degraded and they don't have the ability, because again, this MHC1 basket is protein-based, right? So if you damage the DNA in that location where the instructions are for making the basket, then you may end up losing the basket, so cancer cells may forget how to put that up. Anybody who doesn't have an MHC1 on their surface, viral infected or tumor or whatever the reason is, those guys will be killed by natural killer cells. Natural killer cells also target parasites, and they do this via a process called antibody-dependent cell-mediated cytotoxicity, a super long name that we abbreviate as ADCC. This is how it works. So here's our giant parasite. Okay, so unlike a bacterium or a viral particle or a toxin, a few antibodies is not going to do the trick here. Okay, it's too big. And so instead, what we have is all of the forces come to bear. Antibodies bind to various antigens and help phagocytes and macrophages and so on recognize that this is something that needs to be dealt with. Macrophages can secrete enzymes that cause damage. So can some lymphocytes known as eosinophils that are targeted against parasites. And natural killer cells will also gang up on this guy. So if it's too big to engulf in phagocytosis, you then instead bring in guys that will just shoot holes in it. Okay, that's essentially what's happening here. Lots of flagging and then lots of things coming in that can secrete enzymes that will make holes in this guy. So here's an example of a larval stage of a parasite. And all of these cells are eosinophils, this particular kind of white blood cell that targets parasites. Just totally going after this guy. Okay. So now we have sort of talked at length about different kinds of cells. And let's see how far we have to go, because I want you to have time to do your lab. Oops. Oh, we don't have far to go at all. So may as well finish up. Okay, so cytokines are these one of these things that cells can secrete to talk to each other. Now, where do we know cytokines? We've talked about at least one instance today where cytokines are used. What cell type uses them and for what purpose? T helper cells use them to talk to B cells or other antigen-presenting cells, macrophages, for instance, or dendritic cells, to activate them, to say, yes, I recognize what's in your basket. Cytokines are chemical messengers. They're secreted. They can travel long distances. There are hundreds of them. And if we make too many, then we can end up with something called a cytokine storm. It's a sort of a positive feedback loop that results in a lot of damage. So what kind of cytokines do we have? We have interleukin-1, right? So that is a cytokine that stimulates T helper cells in the presence of antigens, and also it attracts phagocytes. And you may remember that interleukin-1 was also the thing that talks to the hypothalamus, right? In order to do what? What is the hypothalamus's job? Sets the temperature, cytokines talk to the hypothalamus that then releases prostaglandins and resets that temperature higher. Interleukin-2 is another cytokine, and it is important in telling T helper cells that have recognized their antigen to proliferate, that is, go through cell division, make more of them. It also helps B cells to differentiate. It also is important in activation of the CD8 positive T cells, which are what type? CD8 positive are, they're not T helper cells, but cytotoxic T cells. And interleukin 2 is important in activating natural killer cells. We also have interleukin 12 that is important in activating one of the subtypes of T helper cells. And a variety of other ones. So we have chemokines that induce migration of white blood cells. We have tumor necrosis factor that promotes inflammation. That's also discussed in your innate immunity chapter. We have what we call hematopoietic cytokines that are important in talking to the stem cells in the bone marrow to differentiate the right numbers of different kinds of red blood cells and different kinds of white blood cells. Interferon alpha and beta. Who's making this? Interferon alpha and beta. What cells are making them under what circumstance? Remember that whole concept of the local neighborhood watch? Who's making them? What kind of infection? Viral infection, okay? The infected cell makes interferon alpha and beta that diffuses locally and warns the neighbors to start mounting defenses. And the defenses that they mount are inhibiting protein synthesis of viral proteins. Interferon gamma, on the other hand, is something that is a stimulating factor for macrophages. So close to the end of the chapter, just hang in there. All right, so how do we classify the different ways that we acquire immunity? And we'll revisit this in chapter 18. We have what we call naturally acquired and artificially acquired, and just off the top of your head, what do you think the difference is? Naturally acquired could be mom. There's other ways to naturally acquire immunity. Exposure, okay? So anything that normally happens, like you get a cold, or you get born, or whatever it is that's normally part of your life cycle without medical intervention, that's naturally acquired. So what is artificially acquired? Yeah, so we deliberately introduce something into the body on purpose in order to simulate part of your immune response and get there first before you have to go through the uncomfortable actual exposure to the virus or whatever it is. So naturally acquired, we have active and passive, okay? Active being something comes into the body that has an antigen that's recognized by our antigen presenting by our B cells with antibodies, and the body makes antibodies in response to that. It also makes a bunch of specialized lymphocytes in response to that particular antigen. So this is active. This is the whole process. We had to go through the whole thing. The antigen came in, we responded to it, we made antibodies. The antibodies are the result of actual exposure to the antigen. Naturally acquired passive immunity would be what? Did you have to see the antigen yourself? No. You get the antibodies whole. If it's natural, where are they coming from? They're coming from mom, right, during pregnancy. So antibodies that pass from the mom to the fetus or to the infant, so either across the placenta or in the breast milk, those passively acquired antibodies confer immunity, but it's still considered natural. Now, artificially acquired active immunity would be what? We have to see the antigen, but are we seeing the actual virus or the actual bacteria or the actual toxin? We're seeing something like it, okay, that's going to mimic it but not make us sick. So it's vaccination. We put in something that has the antigen that's going to evoke an immune response, but it isn't the actual thing. So it's an artificially acquired active immunity. In fact, technically speaking, if you injected somebody with the virus, which is not natural, and they produced an immune response, technically that's also artificially acquired active immunity. So what is passive artificially acquired immunity? We're introducing something into the body artificially, and what is it? The actual antibodies, okay? So active means we had to see the antigen and make our own antibodies. Passive is we get the antibodies. Artificial versus natural just depends on, did we get it from mom or from the environment, or did we get it from an injection or from some deliberate medical intervention? Is that distinction clear? Okay. All right, so last thing I want to do is just kind of overview of the whole thing now that we've talked about it, just to kind of cement it in your mind. Whoops, that was not intentional. Let's go back for a second. Whee. So we have two branches of the adaptive immune system. We have the humoral, which are what cells are associated with that? B cells, whose job it is to make antibodies. We've got the cellular immune branch, which is primarily T cells, which, although they arise in the bone marrow, where do they mature? In the thymus. What's happening in the thymus? Educating them, specifically educating them to say what? Don't respond to body cells. Okay? Our B cells are looking for, they have antibodies on the surface. What kind of antibodies primarily? IgDs and some IgMs. If they run into that one particular thing they're looking for, then what do? They take it in, they chop it up. What do? Put it in. MHC2. Who reads the MHC2? T helper cells. How does T helper cell recognize that basket? What are the two things it needs? CD4 to bind the MHC2 and the T cell receptor to bind the antigen. If there's a conversation, then what's released by the T cell? Cytokines. We get activation of which cell? B cell and T cell. They talk to each other. Okay, now what does the activated B cell do? It proliferates and differentiates into what? Plasma cells, which do what? Make a ton of antibodies, IgGs primarily, but a bunch of other ones. And then also memory cells. What happens to the T cell? Activated, also proliferates, also generates a subset of its cells are memory cells. Okay. Now let's go over to the T cell side. We have two kinds of T cells, which are helpers and cytotoxic cells. The helpers are using CD4 to read what? MHC2 on what cell types? B cells, all antigen-presenting cells, B cells, and macrophages, and dendritic cells. The T cytotoxic cells are reading what? They're reading the human cells via what? MHC1. How are they binding the MHC1? CD8, right? And what is the consequence of being activated? Turns into what cell type? Cytotoxic T lymphocyte, which makes what protein? Perforin. Perforin, and after it makes those holes, what does it send down the tunnel? Granzyme. And what does granzyme do? It causes the suicide program to begin. Okay, now we know everything we need to know about the adaptive immune system. Give yourselves a hand. Very nice. Okay, so